mango is an entomophilous crop which is having, in spite of having both male and perfect flowers, still insects are essential for the cross pollination in mango, as it was proven by experimentally very long back that insects only play where there is no wind pollination and selfing is very negligible. And since mango is a very highly seasonal crop, so there should be a pollinator and flowering interact. The synchrony is of utmost important, otherwise we may miss, and we may have a lot of flowers but we may not have the fruit set. So considering this, we have conducted over 2-3 years survey for the pollinators across major mango belts of the country, covering from North India and even this Konkan region, Bhubaneswar, and southern parts Bangalore, Chittoor, and including the off-season mangoes of Kanyakumari. Here what we found is majority species composition is more or less same, but some places having some species as a dominant and some having other species. For example, under southern conditions, especially Bangalore conditions, where this Apis florea and Califorid are the major pollinators, and in Vengurla, which is known for this Ratnagiri Alfonso, there we found this mainly stingless bees and syrphids, and we did not find the honeybees. And in Chittoor area, little bee and tetragonal are the important, and in Lucknow, where Dasheri is grown, and there also majority these syrphids were there, and in Kanyakumari also this stingless bees and some other fly species were there. And among the honeybees, sometimes we also find Apis dorsata in large numbers, but when we critically see, they are not coming to the flowers, but they come mainly for the honeydew excreted by the hoppers. So it cannot be considered as a mango pollinator. So this is the diversity of the pollinators on this mango. And among more than 25 plus species recorded, depending on their density and the foraging frequency, these can be considered the very crucial and key pollinators of the mango. Among the bee, Apis florea and tetragonula, and the syrphid Eristalis arborum, and the Califorids, Chrysomia megacephala and Stomorina discolor. These we frequently come across on mango across the varieties. So here when we try to correlate this or study these pollinators in relation to some floral traits, what we like color, density of the panicle and bisexual flower. Here we found color did not have much significant, maybe some very minor variations are there, but there are no significant variations among the panicle color versus this pollinator structure. But panicle density, we found that compact panicles had more pollinator density compared to the this one. And among the varieties, though we have more than 300-400 genotypes we have in our institute, but some representative commercially important varieties we have taken just to see the pollinator among the different varieties. Here also we found some varietal variation is there, and this Alfonso, Ratna and Arkanmal, these were having the high density of the across the species, and some early varieties were having the low. For example, here also flowering time, this mango flowering spans across starts from somewhere November, December and goes up to March. What we found, early maturing varieties had relatively low density of the pollinators compared to the mid and late varieties. And pollinator density also increases, the proportion increases with the extent of the flowering density, and that's why many times the early panicles miss the pollination because the density of the pollinators in the early set of the flowering is low in the mango. And when it comes to how the abiotic factors influence, among this temperature do influence, and here another aspect I was telling, bisexual flowers. These bisexual flowers range from 1 to 35% in different varieties, and here these bisexual flowers in turn influence the pollinator preference. Wherever more the bisexual flowers, where the nectar will be there, so dipterans will be more compared to the honeybees. So that way the high temperature induces the and late flowers will be more bisexual flowers. So we find more dipterans in the varieties with the high bisexual flowers. And the peak activity of the honeybees was in the forenoon hours and coinciding with the anthesis. We studied some our objective was how the temperature influences the, so we have taken the temperature data over the blossoming period. Temperature does impact the pollinators and negative, but here it was the general total pollinators there was a negative correlation, but when we delineated across the species there is a difference, and Apis florea is more affected compared to the dipterans. So here one important thing, we studied this dermal sensitivity of the different pollinators here in terms of this Levin's nest breadth index. In this we see that this califorid Chrysomia megacephala is having high dermal tolerance compared to the our hive honeybees. So it shows that honeybees are more vulnerable to these temperature fluctuations. So they need to be conserved, and we also tried to experimentally prove the contribution of these different species to the fruit set, and we found both Apis florea and mellifera are contributing to this fruit set. And also we developed a protocol for mass multiplying a califorid pollinator for conserving, and also a low cost this fish meal trap also we standardized, so that we advise farmers to erect this in the flowering season, so that we can attract this califorid and conserve the pollinators. So to summarize, Apis florea and califorid and cirripedes are the key pollinators, and here crop phenology as well as temperature influences, and there is a need to conserve this native wild pollinators to sustain this mango pollination. Thank you. And I conducted this much part of this work as under our NICRA project. So I thank ICR and all the our director and organizers. Thank you.
Thank you, Dr. Reddy, for a nice presentation. Now the paper is open for discussion. Maybe one or two questions quickly, please. Any questions?
Sir, what could you say that when temperature is high, which is the effective pollen?
That's what this dipteran pollinators. Because honeybees, being a social insects, they have to do lot of work in their hive when there is a temperature to regulate the temperature. That's why their numbers will be less on the flowers. General density open pollination. To get the yield data and the impact on pollination, we have introduced Florea, Serrana and Califorids separately. Yeah, entire tree was covered and we have put. In nature, this Serrana was coming on few varieties in very low numbers, but Florea is the very number one pollinators under southern, especially Bangalore condition.
Sir, what was the cues pollinators will take for the pollination visible flowers? Apart, since you said that it's not the flower color, and what was other cues, even for the difference or even for the?
Yes, certainly it must be these volatiles only. We have not studied, but certainly this chemical cues only will help with this nectar composition and all.
Sir, when you said that when temperature is rising, bisexual flowers are increasing.
In general, there is some with increase in temperature and also the late flower setting, the proportion of bisexual flowers is more. But in general, the number of bisexual flowers is a varietal character.
So, but these are andromonaceous.
Yes.
So, in that case, the sex ratio, how it will be going to influence in connection of the pollen and the nectar?
See, that's what, in general, it varies from 1 to 30%. Majority of commercial varieties are having 8 to 10% of the bisexual flowers. So, male flowers will produce lot of pollen. That's why honeybees come mainly for pollen, whereas non-honeybees, especially flies, come for the nectar. So, while moving for the nectar, these flies will get in touch, their body will carry the pollen. That we have studied different body parts, how much pollen they carry.
ಹಾಯ್, ನಾನು ನಿಮ್ಮ ಕೃಷಿಕಟ್ಟೆ ಚಂದ್ರಮೋಹನ್. ಸಮಸ್ತ ವೀಕ್ಷಕರಿಗೆ ಧನ್ಯತಾಪೂರ್ವಕ ನಮಸ್ಕಾರಗಳನ್ನು ತಿಳಿಸುತ್ತೇನೆ. ಕೃಷಿಯೇತರ ವಿಷಯಗಳು, ನಮ್ಮ ಕಲೆ, ಸಂಸ್ಕೃತಿ ಮುಂತಾದ ವಿಷಯಗಳ ಕುರಿತಾಗಿ ಸಾಕ್ಷ್ಯಚಿತ್ರಗಳ ಬಿತ್ತರಿಸುವ ನಮ್ಮ ಕಾಯಕಕ್ಕೆ ಬೆಂಬಲವಾಗಿ ನಿಂತ ನಿಮ್ಮ ಪ್ರೋತ್ಸಾಹದಿಂದಾಗಿ ನಮ್ಮನ್ನು ಇಲ್ಲಿಯವರೆಗೆ ಕರೆದು ತಂದಿದೆ. ನಿಮ್ಮ ಪ್ರೋತ್ಸಾಹ ಸದಾ ಹೀಗೆ ಇರಲಿ. ತಮ್ಮಲ್ಲಿ ವಿನಂತಿಸಿಕೊಳ್ಳುವುದು ಏನೆಂದರೆ, ನಮ್ಮ ವಾಹಿನಿಯ ತಂಡ ಸಮಸ್ತ ಕರ್ನಾಟಕದ ಕೃಷಿ ಸಮುದಾಯವನ್ನು ತಲುಪಲು ನಮಗೆ ಹೆಚ್ಚಿನ ಬೆಂಬಲದ ಅವಶ್ಯಕತೆ ಇರುವುದರಿಂದ ನಮ್ಮ ಚಾನಲ್ನಲ್ಲಿ ಜಾಯಿನ್ ಎಂಬ ಬಟನ್ನನ್ನು ಹೊಸದಾಗಿ ಸೇರ್ಪಡೆ ಮಾಡಲಾಗಿದೆ. ಇದರಲ್ಲಿ ತಮಗೆ ಇಚ್ಛಿಸಿದ ಸದಸ್ಯತ್ವ ಅಥವಾ ಮೆಂಬರ್ಶಿಪ್ಪನ್ನು ಆಯ್ಕೆ ಮಾಡಿಕೊಂಡು ನಮ್ಮ ಕಾಯಕಕ್ಕೆ ನೀವು ಇನ್ನೂ ಹೆಚ್ಚಿನ ಮಟ್ಟದಲ್ಲಿ ಬೆಂಬಲ ನೀಡಬಹುದು. ಇಲ್ಲವಾದಲ್ಲಿ ಏನೂ ತೊಂದರೆ ಇಲ್ಲ, ಸಬ್ಸ್ಕ್ರೈಬ್ ಬಟನ್ ಇದ್ದೇ ಇದೆ. ಉಚಿತವಾಗಿ ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ ನಮ್ಮ ಚಾನಲ್ನ ವಿಡಿಯೋಗಳನ್ನು ವೀಕ್ಷಿಸಬಹುದು. ತಮ್ಮೆಲ್ಲರ ಪ್ರೋತ್ಸಾಹಕ್ಕೆ ನಾವು ಸದಾ ಚಿರಋಣಿಗಳಾಗಿರುತ್ತೇವೆ. ಧನ್ಯವಾದಗಳು.